Okay, so it was requested that I create a audio podcast for Figure 1 for a journal club paper. So we're going to look at Figure 1. First we'll look at Panel A, and then we'll look at Panel B, and then I will give you a synopsis of how this information relates to the lecture material. So it kind of will resemble Figure 3, which is that textbook figure. Okay. So looking at Panel A, we want to keep in mind that we have a starvation condition, and then we also have our glycolysis. And so under the starvation condition, we can tell from the figure that we don't have any fructose, we don't have any ATP. But we want to keep in mind there's something else that's lacking under the starvation condition that is present with glycolysis. And so with glycolysis, we need to have glucose present in order for that to happen. And so with our starvation period, we don't have any glucose present. So if we don't have glucose, it's not surprising that we can't have end products from glycolysis. And so ATP would be lacking because we don't have any glucose in order to convert that to ATP during the electron transport system. So we have all that stuff on that chart before that. Because we don't have glucose, we can't bring it inside the cell and phosphorylate it and then convert it to fructose and add another phosphorylation to it. So again, the findings from Panel A are not earth-shattering, but again, if we really think about where these molecules are inside the cell versus outside the cell, it helps us understand the panel. In my last slide for this podcast, I will go over each of these where these different metabolites are found. So there's intracellular phosphate that they're measuring, ATP, the PEP pool, and fructose. So if you have questions about that, just keep on listening. I will go into that. Now, for our next panel, Panel B, one of the important things to keep in mind is that this is based on literature. So this isn't research that they actually did. They weren't sitting at the bench doing this. They went to other people's research, looked at the data they had, and then compiled that into this table. And so what they're measuring here is looking at the fructose levels with the two phosphorylations, and looking to see whether when they change the concentration, or when other groups change the concentration of extracellular phosphate available, whether that changes the amount of fructose biphosphate that it ends up being produced. Now, it's not surprising that if we have more phosphate present, that we'd have more fructose. Now, in all these conditions, glucose has been added, so glucose is available to these cells. And so one of the things I had talked about in Journal Club is to think about what would you do if you could go back and visit this. And so one of the things I probably would think about doing is, what if we added more? The question came up about would eventually you saturate it, and it didn't matter how much extracellular phosphate you had, would you stop producing more fructose? And so if we think about this proposed experiment, what we might find is at a slightly higher concentration from our highest, that we might have more fructose be present. Maybe what would happen if we have an even higher concentration, we would produce more fructose. But then again, maybe what would be more likely is that eventually we'd saturate those enzymes. We wouldn't be able to produce more fructose just because there's more phosphate being present. Maybe by that point we'd used up all the glucose that was added into the system. So again, it's always good to kind of think of what if we did this? What experiment could we do? How could we learn more from what was done by these researchers? Okay, so for this slide, I just want to kind of put all the pieces together. So we drew out on the whiteboard this. It's also kind of removing a lot of the extra information that's in Figure 3. But for Figure 1, they're looking at the phosphate and all the different, or in the major forms that are available for Panel A and in part Panel B. And so in Panel B, they're talking about the extracellular phosphate. panel A, they're measuring the phosphate that moves inside the cell, the intercellular phosphate. They're also measuring phosphate that's in a pool form that's available to use, but is kind of bound so it's not freely floating like the intercellular phosphate. So these two, the intercellular phosphate and your phosphate pool, are available to be used. They're also measuring the fructose, and that's because if you add glucose, glucose is going to move inside the cell. It'll be phosphorylated, converted to fructose, and another phosphate is added to it. So you have fructose biphosphate. So they're looking to kind of see, did the glucose get in the cell and did it move along glycolysis? Now downstream, after many steps in the pathway, after we've continued through and created our pyruvate, we then, through the linking reaction, has made acetyl-CoA, we have the TCA cycle, Krebs cycle, and then we get to the electron transport system. Then we get ATP production. And so they also look at this very downstream product being the ATP. So again, for figure 1, they're looking at those 4 different forms. Now in our other figures, they're also looking at the glucose outside the cell. So they're looking to see whether that decreases, indicating the glucose is going inside the cell. They could have measured intercellular glucose. They didn't. They're measuring the fructose biphosphate again. They measure ATP again, but they also measure this other end product, lactate. The lactate is going to be produced inside the cell and then gets transported outside. So they measure the extracellular lactate just like they measure the extracellular glucose. Okay. So I'd mentioned you get this ATP production. You want to keep in mind that the phosphate that's on the ATP is not coming directly from the fructose biphosphate, that it's during the electron transport system that we get this generation of ATP. And that's because along the way we've produced these energy carriers such as NADH. And so in one of the figures they do look at NADH. They look at NAD and its levels versus the levels of NADH. And so you see this kind of, when one goes up, one goes down effect. And that's because during the electron transport system, your NADH is going to be pumping those hydrogens across the cell membrane of the bacteria. And then as the hydrogens come back in, you're able to get ATP production through the ATP synthase. Okay. So if you do have any additional questions about figure 1 or any of the other figures from the journal club paper, please do not hesitate to contact me, email me, post it on the discussion board. We can talk more about it in class on Wednesday. So thank you.